In the previous unit, we studied about the cell, which is the basic structural and the functional unit of the body. And the examples are the nerve cells and the muscle cells. Now, these cells, when the group of cells combine to perform a specific function, that becomes the tissues. When groups of tissues combine together to perform a specific function, it becomes organs. And when groups of organs combine together to perform specific functions, that become organ system. An organism is nothing but it's the collection of different organ systems. Now, what are the different organ systems? Like circulatory system, digestive system, nervous system, excretory system, they make up the different organ system of animals. And if we talk about just the digestive system, the different organs in that will be stomach, kidney, liver, pancreas, heart. These are the different organs. And then the organs are further made up of smaller tissues like muscular tissue, epithelial tissue, glandular tissues, and the tissues are made up of collections of cells. So you should know these hierarchy and the examples of them. Now, let us talk about digestion. Now, what is digestion? Digestion is the process of breaking down of large insoluble food particles in the food into soluble smaller particles which can then be absorbed in the blood and used up. Now, what are the bigger components which are present in the food? These are carbohydrates, proteins, and fats. The carbohydrates get digested by the special enzymes called carbohydrases into simple sugars. Proteins get digested by proteases into amino acids, and fats are digested by lipases into fatty acids and glycerol. Now, this is what is the structure of the digestive system, and you should know how to label it. So it starts with the mouth and the pharynx, and then there are the salivary glands that releases saliva which softens and moistens the food, and saliva also contains the enzyme amylase that begins the process of digestion of starch to sugars. So you must have seen people says that if you chew the food, it tends to taste sweeter. The reason is that the amylase enzyme, it starts to digest the starch into the food into simple sugars. Then the food is pushed down into this food pipe called esophagus, and then it reaches the stomach. In the stomach, the hydrochloric acid is released. This hydrochloric acid first kills all the infections which comes up with the food, and it also makes the pH acidic so that stomach enzymes can work, because stomach enzymes work in the acidic pH. The digestion of protein starts in the stomach. The food stays approximately 3 to 4 hours in the stomach, and then it goes into the small intestine. When it goes into the small intestine, it gets two secretions. One, from the gallbladder, it receives the bile. Bile is made in the liver and stored in the gallbladder. The bile, it emulsifies the fat and neutralizes the stomach acid pH, which comes up with the food. And then the pancreas releases the pancreatic juice, which contains all the enzymes to finish up the digestion. So all carbohydrates, proteases, and lipase are present in this pancreatic juice. The digestion is finished. In the small intestine, the food gets absorbed into the blood, and then undigested food passes into the large intestine. The large intestine absorbs all the water and converts the undigested food into the semi-solid mass called the feces, and the feces is thrown out of the body through rectum or the anus. So this is a whole process of digestion. If you see this, we can break this digestion process into 5 stages. One is ingestion. Ingestion is the taking in food. It helps with the help of the mouth and the tongue. The food is churned, so mechanical digestion takes place, and the food is mixed with the saliva. Then is the digestion. It starts in the mouth, and stomach, intestine, pancreas, and liver are involved in it, which is the breakdown of large insoluble particles into smaller ones. Then in the small intestine, absorption, the process of digested food are absorbed into the blood. Then assimilation happens in the body cell, where this absorbed food is used for the body processes. And there's an egestion, which is the getting rid of undigested food material, which happen through rectum and anus. Okay, now let us just quickly summarize this entire digestion process. So it starts with the mouth, where food is ingested, mechanical digestion starts, and amylase is mixed, which starts to break down the starch into the sugars. Then it is the pharynx, which is at the beginning of the throat, and both food and air travels to it. Then there's a small flap called epiglottis. Epiglottis is a flap of muscles that closes the windpipe so that the food does not get into the windpipe, and it directs the food into the esophagus. Esophagus is the tube that leads the food into the stomach, and it has some peristalsis movement that pushes the food down. Then the food comes into the stomach, where the mechanical digestion and the chemical digestion starts. The hydrochloric acid is released, and an enzyme called pepsin is released that digests proteins into amino acid. Then the food goes into the small intestine. There, the liver that makes the bile stores into the gallbladder. The secretion of the bile comes into the food that neutralizes the food, and then gallbladder also releases bile, which is stored in the gallbladder. And then the small intestine, where the digestion finish and the absorption starts. And then there's a small organ here called the pancreas, which releases the pancreatic juice. And yeah, pancreas, it is where it produces the pancreatic juice that contains the lipases, proteases, carbohydrases enzyme. Then the food goes into the large intestine, which absorb all the water. Then it is thrown out of the body through the rectum and anus. So you should know all the digestive organ. You should be able to label them in the exam, and you should know the function of each of the parts. Okay. Now then, the very important function is the bile juice. It is a greenish yellow alkaline liquid which is produced in the liver and stored in the gallbladder, and it performs two major functions. You should remember that. One is the neutralization. So the food that comes from the stomach is acidic, and the enzyme of the intestine can only work in alkaline condition. So the bile neutralizes the food that comes from the stomach and makes it alkaline, so that enzyme release in the intestine can work effectively. Next is the emulsification of the fats. For lipase to work, the fat must be broken down into smaller droplets to increase the surface area for the lipase to function. The bile performs this function of emulsification of fat for the efficient working of the lipase. Now, let's see what are the components that we take in with the food. We take carbohydrates, proteins, and fats. Carbohydrates are present in bread, pasta, cereals, and rice. Proteins are present in pulses, chicken, meat, poultry, egg, beans, and nuts. And fat is present in butter, oil, margarine. Carbohydrates are composed of sugars. Proteins are made up of amino acids, and fats are made up of fatty acids and glycerol. And this is what these components get digested into. The enzyme for carbohydrates are carbohydrates. Example, amylase. For protein, it is proteases, and for fat, it is lipases. Carbohydrates are digested in mouth and small intestine. Proteins are digested in stomach and small intestine, and fats are digested just in small intestine. Carbohydrates are the principal source of energy. They are fuel for respiration. They can be storage molecules like starch and glycogen, or structural molecules like cellulose in plants. Proteins are the components of muscles required for growth and repair. They're the components of enzymes, and they are also components of some of the hormones like insulin. Fats are used to insulate the body. They are the reserve source of energy, and they also makes the cell membrane. Okay, so you should know what are the different components of food, their sources, where they're digested, and what is their function in the body. Okay, so now since we are doing digestion, and digestion is chemical, that is, it involves the use of enzyme, you should know what are enzymes. So make sure you remember this definition and this lock and key model, very, very important. So I'll just take you through it. So enzymes are biological catalysts. So I think in chemistry, you remember what are catalysts? Catalysts are those substances that increases the rate of reaction without being used up. So if there's a catalyst which are used in biological reaction, we call them enzymes. So enzymes are biological catalysts that increases the rate of a biological reaction without being used up. Now, what do enzymes do that increases the rate of reaction? They actually provide an alternative route to a reaction, which works by lowering the activation energy. And enzymes are chemically proteins, so they are very sensitive to heat, pH, and temperature. Okay, now this is the structure of the enzyme, and the enzyme has a specific shape, and it has a site called the active site. Active site is a site where substrate molecules bind to it. Now substrate and the active sites, they have complementary shape. Remember to use the word complementary. Most of the time in the exam, students say substrate and active site have same shape. No, that's wrong. It's like a lock and key. The substrate and the active site, they have a complementary shape. The substrate binds to the active site, forms an enzyme substrate complex. The enzyme substrate complex react to form an enzyme and the product, and it disintegrate to give the product back, and your enzyme gets regenerated. So remember, enzymes are highly specific due to the active shape. As the active shape has a specific shape which is complementary to the substrate, so the specific substrate molecule can fit into the active site of the enzyme. Now, what are the factors that affects the enzyme activity? Temperature and pH are one. We'll be discussing that. But the substrate concentration and enzyme concentration also affects the rate of reaction. So as we increase the substrate concentration, there'll be more molecules to bind with the active site. Therefore, the rate will increase at the start, but after that it levels off because all the enzyme active sites will be used up, and then enzymes will become a limiting factor. The same thing work with the enzyme concentration. As we increase the enzyme concentration, there'll be more active sites for the substrate molecules to bind to. But after that, further increasing the enzyme concentration will have no effect because then all the substrate molecules will be used up. So substrate will become limiting. So the graph will level off. Now for the temperature, as we increase the temperature, the rate of enzyme catalyzed reaction increases because the particles gain kinetic energy as per chemistry. They'll collide more the collision, more the chances of reaction. It increases up to a point, and this point is called the optimum. Optimum is the point where the enzyme activity is the highest. But after that, the rate will drop suddenly. Why is that? This is because the enzyme gets denatured. Now, what is denaturation? Now, denaturation is when you heat any specific enzyme due to being protein in nature, their bonds break. So they change the shape. Now, when they change the shape of the active site. So the substrate molecules can no longer bind to it, decreasing the rate of reaction. So denaturation is change in shape of the enzyme with a change in pH or the temperature. The same effect happens with the pH. As you increase the pH, it increases the rate of reaction up to the optimum, and after that it drops. It doesn't drop that suddenly like temperature, so we make like a bell-shaped curve. And then it again drops. Why? There's a denaturation because the shape of the enzyme changes. Now for the temperature, the optimum is around 37 to 40 degrees because that is the normal body temperature. But for the pH, different enzyme has different optimums. Like stomach enzymes, they work best in acidic condition, and the intestinal enzymes, they work in the alkaline condition. Okay, so I hope this graph is clear to you. You should be able to draw this graph in the reaction in the exam, or if this graph is given, you should be able to explain all this trend, why there's an increase, what is an optimum, and why there's a drop. And I hope you will remember it. Now let's move on to a circulatory system. The question is, why we need a circulatory system? And what is a circulatory system? So circulatory system is a system that provides the body with oxygen, nutrients, hormones, or all the essential things that a body needs to survive. And it also gets rid of the waste products. But why do we need a circulatory system? We need a circulatory system because we need constant supply of all these nutrients. And circulatory system consists of three components: a pumping organ, an organ that pumps the transporting system, transporting medium. Transporting medium is the blood, which has all the things that a cell's required. It is pumped by a pumping organ called the heart, and the blood is taken from one cell to another, or from one organ to another, to the system of vessels, which is arteries, veins, and capillaries. So now we'll be looking over what is blood, heart, and these arteries, veins, and capillaries. Now, if you talk about the blood is made up of blood plasma and blood cells. So if you see blood, we centrifuge the blood, then the blood cells are heavy, they collect at the bottom, and then there's the blood plasma. Blood plasma makes up 55% of the blood, and it is the liquid component of the blood, and it has all the things that a cell requires, like glucose, amino acids, hormones, waste product, carbon dioxide. And then the blood cells, they perform specific function, and there are three types of blood cells: red blood cells, white blood cells, and platelets. The function of the red blood cell is to transport oxygen all around the body, and it's a special cell. Why it's a special cell? Because to carry out these special functions, it has special adaptations. Like, they are in the form of biconcave disc, which increases the surface area for oxygen transport. They have no nucleus, so that they can have more room for the oxygen, and they contain a pigment called the hemoglobin, which binds with oxygen and carry it all around the body. Then you have the white blood cells. White blood cells are the soldiers of the cell. They protect the body from infections. They are made up of two types of cell: lymphocytes, they produce antibodies, and phagocyte, which engulfs the pathogen. And then we have the platelets. They are involved in blood clotting. They have fibrin proteins, which form mesh around the blood. and prevents the excess blood from flowing out by forming a clot. So I hope you will remember what are the different cells and their function, and what is the function of the blood plasma, and what are the different components of the blood. Okay, so now we will talk about the heart, but before that we should know why we need a circulatory system. We need a circulatory system because we have low surface area to volume ratio, we are metabolically active, and we are multicellular. Now, low surface area to volume ratio because we have a bigger size. As the size increases, the surface area to volume ratio decreases. And at such a low surface area to volume ratio, diffusion alone is not effective to meet the growing need for the body. Therefore, we need a circulatory system. Now, being metabolically active, we need a constant supply of oxygen, glucose, and nutrients. And at the same time, the waste product is also made at a greater level. So we need a constant removal of waste products. So we need a circulatory system to do that task. And being multicellular, we have lots of cells which are deep inside. So diffusion distance is too large. So we need a circulatory system to transport substances deep inside the cells as well. So therefore, we have this circulatory system, and the circulatory system we have is double circulation. Now, let me explain you how this double circulation works. Now, in our circulation, we have a lungs and the heart is 4 chambers. The top chambers are the atriums, and the bottom is the ventricle, and our right is the heart left. So this is the left atrium, left ventricle, right ventricle, and the body. Now, how does the circulation start? The lungs gives oxygen to the blood, and the oxygenated blood from the lungs, it starts flowing to the left atrium. The left atrium then pushes the blood to the left ventricle, and then left ventricle pushes the blood out, and it goes to all parts of the body. The body takes oxygen and all the required nutrients from the blood, and then it releases carbon dioxide into the blood, and the blood becomes deoxygenated. This deoxygenated blood from the body is pumped back into the right atrium. The right atrium takes the blood to the right ventricle, and the right ventricle then takes it back to the lungs so that it can pick some more oxygen, and the cycle continues. Okay, now if you see the circulation, it is consist of 2 circuits. One, lung to the heart and heart to the lung, and that is known as pulmonary circulation, and heart to the body and body to the heart, that is known as systemic circulation. And both of these circulation together make a double circulation. So that is one of the definition of double circulation, that it consists of 2 circuits, a pulmonary and the systemic. The second way to describe this double circulation is, this is circulation in which the blood is pumped to the heart twice in one complete cycle. So in one complete cycle, the blood is pumped to the heart twice. It involves 2 circulation, pulmonary, which is between lungs and the heart, and systemic, which is the circulation between heart and the body. The double circulation makes the circulation more efficient by preventing the mixing of oxygenated blood, and helps to alter the pressure from the different chambers of the heart. So here you can see in double circulation, the oxygenated blood is all to the left side and deoxygenated blood to the left side, and the heart can modulate the pressure. When it is sending the blood to the body, it can increase the pressure, and when it is sending the blood to the lungs, it can decrease the pressure, and that can only be done as it has a double circulation. Now let us label the vessels. Now, if to label the vessels, always remember anything that gets the blood to the heart will be the vein, and anything that takes the blood away from the heart is an artery. So in the first case, the blood is coming to the heart from the lungs. So this has to be a vein. And since it is the vein associated with the lung, so it has the word pulmonary vein associated with it. So the blood comes from a pulmonary vein to the left atrium. Then it goes to the left ventricle, and then from the left ventricle, it goes to the body. So blood is going away from the heart. So this has to be an artery, and it has to be a largest artery, as it is giving the most difficult task of sending the blood to all parts of the body. So this is aorta, which is our largest artery. Then the blood comes from body to the heart. Again, it has to be a vein. So it's a vena cava. And then from the right ventricle, it goes back to the lung. So it has to be the artery. And since it is associated with the lung, so it is the pulmonary artery. So you should be able to draw this circuit and label all the vessels. And I hope this is clear to you. Next is we have this heart. Heart is a pumping organ, works all day and night. It has 4 chambers. Atriums are the top. So this is left atrium. This is right atrium. The ventricles are at the bottom. So this is left ventricle, the right ventricle. Ventricles are doing the biggest job as you're pumping the blood to a greater distance. So you can notice that atrium has a thin wall and ventricle has the thickest wall. And as compared to left ventricle and right ventricle, left ventricle has to send the blood to all parts of the body. So it has to send the blood at a very high pressure. So it has the thickest wall. Okay, and then the left atrium receives the blood from the pulmonary vein. It goes to the left ventricle. The left ventricle sends the blood to all parts of the body. So that's the aorta. The blood travels all parts of the body and come back to the right atrium through the vena cava, and from the right ventricle the blood pump back to the lungs via pulmonary artery. So you should know the structure of the heart and the labeling and the functions of all the parts, and I hope this is clear to you. Now, if you remember that in a circulatory system, we need the blood vessels that carries the blood all around, and there are three types: arteries, the capillaries, and the veins. The capillaries connects arteries and veins, and what you can see in arteries and vein, the lumen of the vein is bigger and the lumen of the artery is smaller. On the other hand, the artery is surrounded by a thick muscular tissue which is not present in the capillaries, and veins have thin. So let's tabulate the difference between them and make sure you remember all these differences and able to write in the exam. Arteries carries the blood away from the heart, veins carries the blood to the heart, capillaries connect arteries and veins. Arteries blood flow at a high pressure, in veins it flow at a lower pressure, and capillaries flows at a lower pressure. So to withstand high pressure, the arteries have thick muscular elastic wall, veins have thin walls, and capillaries are one cell thick. Arteries have a narrow lumen, veins have a wider lumen, and capillaries have a very small lumen. As the blood flows at a very low pressure in veins, there are chances that the blood can backflow. So veins have valves to prevent the backflow of the blood. Arteries and capillaries do not have valves. Arteries generally carry the oxygenated blood except the pulmonary arteries. Vein carries deoxygenated blood except the pulmonary vein. And the capillaries carries both oxygenated and deoxygenated. So I hope you are able to spot by the diagram whether it's in arteries, veins, and capillaries with their function. Okay, now since we have discussed the heart, there are a lot of problems that can come up with the heart, and they are known as coronary heart diseases. Now, what are coronary heart diseases? If there's a problem in a coronary artery. What's a coronary artery? It is an artery it's the branch of the aorta that supplies the blood to the heart. And if these arteries become narrow, why they become narrow? You can see this is a normal coronary artery, and in the coronary artery there is a plaque that is deposited. What is a plaque? It is a fatty materials like cholesterol which get deposited. It narrows the artery. The narrowed artery reduces the blood flow to the heart. The blood flow to the heart is reduced. The heart do not get enough oxygen. It can cause heart pain, chest pain, and heart attack. So this is what is the plaque deposition that happens in a coronary artery. Now, how we can improve that? We can insert a stent. A stent is placed which has a balloon with it into the blocked artery. The balloon is inflated and the balloon opens the blocked artery, and a mesh stent made up of metal is placed which holds the narrow artery and prevent it again from collapsing. And people are also given the medicine called the statins, which lowers the blood cholesterol and prevents the further deposition. So this is one of the way, and this is also known as angioplasty, in which a balloon or the stent is used to open the blocked artery. But there are cases where the stent cannot be performed due to maybe the blockage is too much or some of the problems to be done a patient can have with the stent. Then in that case we do a bypass. What's a bypass? See, you can see that this is a blocked artery. We give a graft, and this graft bypass the blocked artery. Okay, so there is a blockage in the coronary artery. An artery graft is stitched to the coronary artery, so the blood, rather than flowing through the blocked artery, it flows to the graft and it resumes the blood flow. Okay, now there are another problems as well apart from this that can be associated with the heart. These problems are like a leaky valves. A person can have leaky valves. Maybe by time that the blood flows at a very high pressure, so the person can develop leaky valves. So due to increased pressure of blood flow, the valve starts to leak. For that, we have valve replacement, where the valves made up of metals or polymers are placed, or the biological valves from the animals can be used. At times, the person get irregular heartbeat. Why? Because the heart is controlled by the groups of cells present in the left atrium called the pacemaker, which regulates the heartbeat. If there's any problem with that, there's an irregular heartbeat. So we make electrical devices called artificial pacemaker, which takes over the function of the natural pacemaker, and they regulate the heartbeat. Then, apart from all this problem, the person can have a greater damage to the heart or heart failure. In that case, we need a heart transplantation or artificial heart, which is not that successful, but the research is going on, but heart transplant can be done. So these are all the non-communicable diseases, or you can say the problems with the heart, and the proposed treatment for the same. Okay, now after the circulatory system, we come to the human gas exchange system called the respiratory system, which starts with the nose, and then the air goes into the windpipe. The windpipe is known as the trachea. The trachea is covered by the ring of cartilages that prevents the trachea from collapsing, and then the trachea bifurcates into two. These two bifurcations are known as the bronchus. These two bronchus enters the lungs. In the lungs, the bronchus gets further branched into the bronchioles, and at the tip of the bronchioles, we have greater air sacs called the alveoli, where the gas exchange takes place. And then there's a dome-shaped muscular organ called the diaphragm, which goes up and down to regulate the chest volume for inspiration and expiration. Now, what is the function of the respiratory system? Breathing system is to breathe. Breathe is taking in oxygen and giving out carbon dioxide. Now, for the air to come in, air always move from high pressure to a low pressure. So since the air coming in, the pressure inside the lung is low. For expiration, the pressure inside the lung is high. If the pressure is low, the volume of the lung should be high, and the volume of the lung should be low in expiration. How these volume increases in inspiration? It is the muscles, the ribs muscles, they contract and the rib moves outwards. In expiration, the ribs relaxes and the rib move downwards. The diaphragm, it flattens, which increases the volume and the air moves in, and expiration, the diaphragm domes up, which decreases the volume and the air pushed out. So you should know what is the mechanism of inspiration and expiration, and what changes takes place inside the lungs or the ribs for inspiration and expiration to take place. For gas exchange, if you remember, we talked in cell biology, there's an alveoli which has a greater surface area by having a special shape and being millions of them. They have a shorter diffusion distance by being one cell thick, and they maintain a steep concentration gradient by having a rich blood supply. By all this factor, alveoli has adapted themselves for an efficient gas exchange. Now we have discussed organization in animals. Now let's discuss organization in plants. The plant cell is there. We discussed the plant cell in the cell biology video. Then the plant cell get organized into tissue, xylem, phloem, these palisades and spongy mesophils. These are the tissues. You can see sponges are the ones which have greater air spaces between them but are photosynthetic. The palisade cells are arranged next to each other, which are also photosynthetic. In the organ system, we have a water transport system and the food transport system. Water transport system consists of xylem and the roots. The food transport system consists of the phloem and the leaves, and all of this make a plant organism. Okay, now what is a transpiration? Transpiration is the loss of water from the surface of the leaf in the form of water vapor, and this happens to a stomata. So stomata are these air spaces or air pores present on the surface of the leaf, which consist of stomatal pore and these cells, which are known as the guard cell. The guard cells open and closes, which controls the water movement, and the gas exchange also takes place to the stomata. Now this transpiration makes the water uptake or the water transport system. How? Water is lost from leaf pore in the form of transpiration. This decreases the concentration of the water, so the water from the neighboring cell moves in. Now, as the water from the neighboring cell moves in, that decreases the concentration of water in that area. So it takes water from the neighboring cell, and then from the neighboring cell, which makes a long continuous column, and eventually it forces the water from the soil to comes into the root, and then the xylem carries up due to transpiration stream. Okay, now what are the factors that affects transpiration? It is temperature, light, wind, and humidity. With increase in temperature, rate of transpiration increases because the particles gain kinetic energy, they start to evaporate faster. With light, rate of transpiration also increases because remember, as the photosynthesis will increase, the transpiration will increase, because as the rate of photosynthesis increase, the matter will increase to take in more carbon dioxide, which increases transpiration. Wind, as the wind speed increases, it moves all the saturated air, increasing the concentration gradient of the water vapor, increasing the rate of transpiration. Humidity, as the rate of transpiration decreases with humidity. Why? Because humidity will make the air saturated with water vapor, decreasing the concentration gradient, so the diffusion rate of water will slow down. Okay, now how do we measure the rate of photosynthesis or transpiration? I think you must have done this experiment. This is a potometer. A potometer, there's a glass tube, there's a reservoir of water, and there's a beaker, and there's an air bubble. As the water uptake, as the plant will lose water, it will try to uptake this water, and this bubble will move. You will use a scale and measure how much the bubble is moved. The movement of bubble is an indicator of water movement, which in turn shows the rate of transpiration. So we assume all the water that it is uptaking is equivalent to the rate of transpiration of water. Okay, now since transpiration is a necessary evil, why? Because transpiration is required for the uptake of water, but it can also cause the plant to unnecessarily lose the water and can wilt, they can droop. So the plant has done an adaptations to limit water loss. They are covered with the cuticle layer, which is waterproof and prevents the rate of transpiration. They have stomata on the lower surface that reduces the rate of transpiration. Some plants' stomata opens at night and close during the day. In some plants like cacti, the leaves are reduced to spines that limits the rate of water loss. And some plants have sunken stomata, where the stomata is in pits, and they have the humid air around it that prevents the rate of transpiration. Okay, so I hope this unit is clear to you. Now it's the time to test yourself. In this unit, we have seen a lot of key terms. So you should be able to explain these key terms. So these key terms are cell, tissues, organs, organ system, digestive system, esophagus, stomach, liver, pancreas, small intestine, large intestine, substrate, active site, carbohydrates, protein, fats, enzyme, catalyst, denaturation, amylase, protease, lipase, bile, emulsification, plasma of the blood, red blood cell, white blood cells, platelets, plasma, arteries, veins, and capillaries, double circulatory system, coronary artery, coronary heart diseases. Now, you must remember from your key stage 2, 3 that photosynthesis is the process in which the light is used up by the plant, and it also uses carbon dioxide. It combines it with the water taken from the soil, and the plant has chlorophyll that traps the light, and that converts all these into sugar or carbohydrates and releases oxygen as a byproduct. So this is what is the process of photosynthesis. So it is a process by which green plants prepare their own food by using water, carbon dioxide in presence of sunlight to form glucose and oxygen. And you need to remember this equation. This is very important. CO2 and water in presence of light, and you can also write chlorophyll if you want, to form sugar and oxygen, or glucose and oxygen. If you can remember the word equation, that would be great. So it is carbon dioxide plus water, H2O gives C6H12O6 plus O2. Remember, you have to put 6 everywhere except in the glucose. Okay, now why only leaf can photosynthesize? Why not roots? The reason for that is the presence of chloroplast. The leaves have chloroplast, which contains chlorophyll in it, which is a green pigment that can trap light energy from the leaves, for the leaves, and helps them to photosynthesize. Next is, if you remember from the energy changes video for chemistry, we discussed what is an endothermic and an exothermic reaction. So if there's any reaction that takes in heat or energy, that's an endothermic reaction. Now you can see in this, photosynthesis is taking light energy, hence it is an endothermic reaction. In terms of bond making and breaking of chemistry, you can also explain endothermic as it requires more energy to break the carbon dioxide and water bond. Then it releases energy in forming the glucose and the oxygen bond. So you need to remember that photosynthesis is an endothermic process. And why is it endothermic? You should know that. Now, in order to do maximum photosynthesis, you can see that the leaf has come up with a very great structure that makes it possible to photosynthesize in the best way. What are the adaptation a leaf has for photosynthesis? If you see the leaf, they always lie flat, they are thin, and they have veins in them. So the veins ensure the quick delivery of water and the minerals. They lie flat so that the surface area is the maximum for the absorption of the light. On the other hand, they are packed with chloroplast and chlorophyll that can trap maximum light for photosynthesis. They require carbon dioxide and they leave oxygen. So for a gas exchange, they have air spores in it called the stomata. The stomata have the guard cells which opens and close to trap and exchange the gases. Next, the photosynthetic cells, can you see, they just lie at the surface so that they can use the maximum light possible for photosynthesis. So they can ask you in the exam what are the different adaptations a leaf has for maximum photosynthesis. So make sure you remember them well. Okay, next is what are the factors that affects the rate of photosynthesis? Now, the factors that affects the rate of photosynthesis, you can easily see these are the factors which are actually affecting the photosynthesis. Okay, so if you see oxygen is required, carbon dioxide is required, water is required. So all these factors, they affects the rate of photosynthesis. Okay, so if we see here, these are the factors which are affecting the rate of photosynthesis. It is the light, carbon dioxide, temperature, and the pH. Okay, now how does light affect the rate of photosynthesis? As the light intensity increases, the rate of photosynthesis increases up to a point and then it levels off. Why is that? Because further increasing a light up to a threshold can make other factors limiting. Like if the light increases too much, maybe that can increase the temperature. So high temperature can then be a limiting factor. The same thing happens with carbon dioxide. If the concentration of carbon dioxide increase, the rate of photosynthesis increase up to a level, then it levels off. Now, why it increases up to the level? Because we cannot increase carbon dioxide beyond a certain point. Why? Because if we increase carbon dioxide beyond a certain point, it can affect the pH and then the pH can become a limiting factor. Now, for the temperature, the graph looks a little different and you need to remember this graph and explain this graph. As the temperature increases, as per chemistry, the particle gains more kinetic energy, they collide more, increasing the rate of reaction. And that's what we can easily see from the graph, that as the temperature is increasing, the rate of photosynthesis is increasing. Now, as it reaches 40 degrees, there we get the maximum rate of photosynthesis, and this point where we get the maximum rate is known as the optimum temperature. Now, as it reaches the optimum temperature, then it suddenly drops beyond that. Why is that? Because the temperature greater than 40 degrees is not good for the enzymes. You need to remember at this point, you have to use this word, the enzyme gets denatured. Now, what is the denaturation of the enzyme? With high temperature, the proteins, enzymes are generally proteins, with sensitive to temperature, the proteins change its shape or the enzyme changes its shape. As the enzyme changes its shape, it is no longer effective as the substrate molecules can no longer bind to it, and that decreases the rate of photosynthesis suddenly. Okay, whereas in pH we get a little bell-shaped curve and it works the same way. The rate increases up to the optimum and then it drops. Why? Because the same thing, the high pH, it changes the shape of the enzymes which make them non-functional. Okay, so there is a law of limiting factor, that any factor which is present in the least amount, either carbon dioxide and water, that will affect the rate of photosynthesis, and that factor is known as limiting factor. So in order to get maximum rate of photosynthesis, what we should do, we should control these limiting factors, and that we easily do in a greenhouse that we'll be checking out next. Now, you must have done this experiment in school. How do you measure the rate of photosynthesis? We take a aquatic pondweed like Elodia hydrilla. We insert it into water, okay, and then we measure the temperature of the water, and then we bring the light intensity and we will see the bubbles coming off. The bubbles coming off is of oxygen, which indicates the photosynthesis is taking place. More the bubbles, greater is the rate of photosynthesis. So you can test various factors affecting photosynthesis by this experiment. If you want to check the light intensity, you can bring the light source near this and count the bubbles. Otherwise, you can take these light source a little far away and then again count the bubbles. And the greater the number of bubbles, greater is the rate of photosynthesis, and that can give you the indication about the rate of photosynthesis. So you can test various factors like temperature, water concentration, the intensity of the light, and you can plot the graph and see how the rate of photosynthesis is affected. Okay, and in most of the cases you will get this graph. With light you will get this graph, carbon dioxide you will get this graph, with temperature you will get this graph. You need to remember all these shapes and the explanation for the same. Now the question is, the glucose that is made in photosynthesis, what happens to that glucose? What is the fate of this glucose? Now this is the slide that summarizes what are the different ways in which the glucose is used up. The first basic way is it's the fuel for the cells. It's the fuel for the living organism. So this glucose is broken down during respiration and give energy to the plant to carry out their daily activities, grow and reproduce. The excess glucose is stored as starch, and starch is used up as a reserve food whenever it is needed by the plant. Now they'll ask you why we store the excess glucose as starch. Why we not just store it as glucose? The problem is the glucose is soluble in water, and as it's soluble in water, it can disturb the osmotic balance and can cause unnecessary osmosis of water. On the other hand, starch is insoluble complex molecule. It's a bigger molecule. It can take in large amount of glucose molecule. It can store large amount of glucose being compact, and the basic importance of that is it is insoluble. So as it is insoluble, you need to say it does not disturb the osmotic balance of the cell. Next is, you know that the plant has a cell wall made up of cellulose, and cellulose is also complex carbohydrate. So glucose can be converted into cellulose, which can provide shape and support to the plant. The glucose, if it combines with nitrates and other mineral ions, it can form proteins, and the glucose can also be converted into lipids, fats and oils, which can be stored as an emergency food reserve, or they can also be stored in the seed, which can provide the energy store for germination. So you should know what are the various ways in which the glucose is being used up in a plant cell. Now there's a test for starch. If you remember from your school chemistry or biology experiment, if there's a starch present, you add an iodine solution and the iodine solution turn blue-black. Now I'll take a leaf and I'll cover one leaf with a small black tape in the middle, and then I'll put it into the light. And then if I add iodine solution to this leaf, what I would observe that the part which was exposed to light will turn blue-black, because this was the part that got the privilege of getting photosynthesized and make starch, whereas the one which was covered didn't get any light, so it didn't get any chance to photosynthesize, so no starch is being made, so it didn't turn blue-black. So you should remember this iodine test for starch, and you can even test it on variegated leaves. What are variegated leaves? Which have different colors. Okay, so wherever there's a green color, that part will turn blue-black, the rest of the part will remain as the color of the iodine. Okay, so next is a greenhouse. Greenhouse, I think you must have seen in some nurseries, they cover and grow the plant in these covered conditions. The purpose of the greenhouse is it controls all the limiting factors to provide the maximum yield of photosynthesis. The limiting factors like temperature, light, carbon dioxide are manually controlled. It works on the principle of greenhouse effect, and it is beneficial for the countries like UK, where the temperature and light is not that much during the winter season. So you can put them in the greenhouse. The greenhouse allows the light to enter but didn't allow it to escape. So thereby the temperature is maintained and the light intensity is maintained. Otherwise, the farmers use bulbs and then cylinders for carbon dioxide and artificially control them to get the maximum possible yield of photosynthesis. Because the yield of photosynthesis can even vary during different times of the day. During morning, the light intensity is the limiting factor. When it comes to the noon, the light intensity is highest, but then the carbon dioxide closes to prevent transpiration, then carbon dioxide becomes a limiting factor. But we can control all these factors in a greenhouse and get maximum rate of photosynthesis, especially for those crops which are economically very important for us, like rice, wheat, and all those crops. Okay, so I hope the concept of photosynthesis, factors affecting photosynthesis, how we can maximize photosynthesis is clear to you. Now let us do the reverse of photosynthesis, which is respiration. Now, respiration is a process of breakdown of food to release energy. And it happens via two mechanisms. One is the aerobic and second is anaerobic. Aerobic, as the name suggests, air. So it happens in the presence of oxygen. Anaerobic takes place in the absence of of oxygen. Aerobic releases more energy as the food is completely broken down. Anaerobic releases less energy as the food is not completely broken down. Aerobic respiration takes place in mitochondria, and it is the opposite of the photosynthesis, whereas the anaerobic respiration takes place in this cytoplasm. Now, you should know the word equation for both aerobic and anaerobic respiration. So, for the aerobic respiration, it's the opposite of photosynthesis. So, glucose plus oxygen gives carbon dioxide and water. So, C6H12O6 plus 6 oxygen gives 6 CO2 and 6 water. For anaerobic respiration, there are two types, one in plants and animals. The anaerobic respiration in plant is fermentation, whereas glucose breaks down to form ethanol and carbon dioxide. And this is a very important process that takes place industrially to produce alcohol from yeast. So we take yeast and we give it the glucose, and the yeast has the enzyme which converts glucose into ethanol. The second importance for this reaction is it produces carbon dioxide, which is Baker's friend. So yeast is known as Baker's friend because we take yeast converts all the sugar and makes carbon dioxide. This carbon dioxide rises and makes the dough soft. Whereas in animals, the anaerobic respiration takes place in muscles where glucose is converted into lactic acid. So you should know the difference between aerobic and anaerobic respiration. You should know the equations of all of them and what is the difference in anaerobic respiration in plants and animals. Okay, so I hope this is clear to you. Now, most of the time students confuse and say breathing and respiration is the same. But that's not right. Breathing is a physical process. Breathing involves no chemical reaction. It's just taking in oxygen and giving out carbon dioxide. Whereas respiration is using this oxygen, combining it with the product of digestion, which is glucose. So it is a reaction between glucose and oxygen to form carbon dioxide and water, whereas breathing is just taking in oxygen and giving off carbon dioxide. Breathing does not involve enzymes. Respiration do involve enzyme. Breathing is a physical process. Respiration is a chemical process. Breathing releases no energy. Respiration is an exothermic process. It releases energy. Breathing takes place outside the cell. Respiration is a reaction that takes place within the cell. Now, if you remember, we just did that photosynthesis is endothermic. So respiration is exothermic. Exothermic means the energy released in making carbon dioxide and water is more than the energy taken in to break glucose and oxygen. Now, I hope you remember the site of respiration, that is mitochondria. Now, if you see the inner membrane of the mitochondria is folded. Now, these folding is known as cristae. Cristae are the folding of the inner membrane of the mitochondria. They increases the surface area for the attachment of enzyme. Therefore, mitochondria is well adapted to carry out respiration. Okay. Now, the question is, what is the importance of respiration? Why do we need energy? Now, we need energy for these reasons. We need energy for movement. We need energy for transport of substances. We need energy to maintain the body temperature. We need energy to do catabolism. Catabolism is breaking down reactions of the body. We need energy for anabolism. Anabolism is making bigger molecules in the body. So we need energy for the muscles to contract, because when the muscles will contract, then only we'll be allowed to move. So muscle cells are responsible for movement, and they are well adapted to do respiration. Therefore, they have loads of mitochondria, and they also store glycogen, which is the fuel for respiration. Next is for transport. You know that for active transport, we need energy because it is a movement of substances against the concentration gradient. So we need energy for the transport via active transport. Now, if you remember, respiration produces heat. So if we need to maintain a higher body temperature, the respiration rate has to increase. So thermoregulation, that is respiration produces heat to maintain the body temperature. We need energy. We need energy for digestion. We need energy to break down carbohydrates into sugars, proteins into amino acids, fats into fatty acids and cholesterol. On the other hand, we need anabolism. We need the glucose to be converted into cellulose in the plants. The energy is needed to convert glucose into glycogen into animals. The energy is needed to convert glucose into protein, fats, lipids, and oil. So for all these processes, we need energy. This question can come in the exam. What are the various ways in which the body uses energy? So you should remember all these factors. Okay. Next is a very important concept. What changes do you see in your body when you exercise? Now remember, whenever you exercise, the demand for two things suddenly increases in your body. This is the demand of oxygen and the demand of glucose, because we need energy, and glucose and oxygen are the raw materials for energy. So for having more oxygen, definitely, where do we get it? By breathing. So your breathing rate increases. So the breathing rate increases so that more oxygen is inhaled to meet the demands of the oxygen by the muscles. On the other hand, when you're exercising a lot, as your respiration rate increases, the carbon dioxide is built up at a greater rate in your cells. So that also needs to be expelled out of the body, and that again happens via increase in breathing rate. Next is the glucose is transported into the body via blood. Okay, and the blood is pumped by a heart. So to meet the demand for increasing glucose, your heart rate increases. It pumps more blood to the muscles, increases blood supply, increases the supply of both glucose and oxygens via blood to a muscle cell. Okay, and your body stores excess glucose as glycogen. So as the demand for glucose increases, the glycogen needs to be broken down back into the glucose, and this process is known as glycogenolysis. So the glycogen breakdown happens, and that breaks down happens into your cells, in the liver cells, to convert the lactic acid back into the glucose. The glycogen, sorry, back into the glucose. So that happens in your muscles. The glycogen, excess glycogen, gets converted into glucose. Okay. Now, have you ever seen that even after you have exercised, you have stopped exercise, your breathing rate is still high? What is the reason for that? Now, the reason for that is during exercise, due to less demand, less availability of oxygen, the body switches to anaerobic respiration. Now, if you remember, the anaerobic respiration in animals converts glucose into the lactic acid. Now, this lactic acid, when developed in greater quantity, can cause muscle fatigue, redness, swollenness of the body. But this lactic acid cannot be stored. It needs to be broken down back into carbon dioxide and water with oxygen. So your body has a depth of oxygen that even after you have stopped exercising, your body breathing rate will continue to high until this lactic acid gets broken down into carbon dioxide and water. So this is what happens after exercise. The oxygen depth, which is the extra oxygen needed by the body after the exercise to recover, is the oxygen needed to break down lactic acid into carbon dioxide and water. And this is why your breathing rate increases until all the lactic acid is broken down back into carbon dioxide and water. So make sure you remember this phenomenon of oxygen depth. Now, what is metabolism? Metabolism is the sum of all the reactions of the body, and we can categorize the metabolism into two categories. Catabolism is the breakdown reactions of the body, where breakdown of glycogens, proteins, lipids, respiration, that are all breakdown reaction. Anabolism is the synthesis reaction in which the bigger molecule is formed from the smaller ones, which is the synthesis of cellulose, starch and glycogen, synthesis of fats and oil, synthesis of proteins. Okay. Now, the last thing we need to do, liver. Why we are studying liver? Because liver is a very important organ involved in metabolism. It is doing a lot of synthesis and the breakdown reaction. Now, it is involved in detoxification, breakdown of the blood cells, breakdown of the lactic acid, and the breakdown of the harmful substances for excretion. So when we take in excess alcohol, that is broken down by your liver. When the blood cells are old, they are worn out, they are broken down in the liver. The lactic acid that is produced by anaerobic respiration, that is also converted into carbon dioxide and water by liver, and that is the oxygen depth. And the harmful protein, the proteins cannot be stored in the body, so it is the liver that converts it into urea, which is excreted by the kidney as urine. Okay, so I hope you will remember all the functions of the liver. Okay, so now in this topic, just to summarize, we did what is photosynthesis and respiration, and this is the difference between photosynthesis and respiration. Photosynthesis takes place only in plant cell. Respiration occurs in all living cell. Photosynthesis is endothermic, respiration is exothermic. In photosynthesis, oxygen is produced. In respiration, oxygen is used up. Photosynthesis takes place in chloroplast. Respiration takes place in mitochondria, or if it is anaerobic respiration, that will take in the cytoplasm of the cell. The photosynthesis is an anabolic reaction where glucose is made. Respiration is a catabolic reaction where glucose is broken down. Photosynthesis is dependent on light. Respiration is not that light dependent. Okay, so I hope this topic is clear to you. Now, this is the time to test yourself to see if you remember the key terms that we did in this topic. We talked about photosynthesis, which is the process by which green plants prepare their own food. Respiration, breakdown of food to release energy. Limiting factors is the factor that limits the rate of photosynthesis. Endothermic reaction is the reaction that takes in heat. Exothermic reaction is a reaction that releases heat. Glucose is a product of photosynthesis and fuel for respiration. Starch is the storage carbohydrates in plants. Glycogen is the storage carbohydrates in animals. Aerobic respiration is the breakdown of food in presence of oxygen. Anaerobic is the breakdown of food in absence of oxygen. In fermentation, anaerobic respiration in plants that produces ethanol and carbon dioxide. Greenhouse is a glass or a plastic house to control the limiting factor and increase the rate of photosynthesis. Metabolism is the sum of all catabolic and now, what exactly is homeostasis and why is it important? Now, homeostasis is the process of maintaining the constant internal environment. Now, the question is, what is constant internal environment? Constant internal environment is maintaining a constant temperature, pH, blood glucose, and salt and water level. Now, when we maintain a constant temperature, that is known as thermoregulation. Maintaining salt and water level is osmoregulation. So, maintaining all these factors is what is homeostasis. Now, how this homeostasis is achieved? This is achieved by two systems working in your body, the nervous system and the hormonal system. Now, why this constant internal environment is important? This constant internal environment or homeostasis is important for the enzymes, as it is the enzymes which are controlling all the reactions and metabolism of the body, and they need optimum conditions to work. If the temperature increases or the pH level increases, it can change the activity of the enzyme and it can affect your body reaction. So it is very important to maintain the homeostasis. Now, whether it is a nervous system or the hormonal system, how it works, it's the whole hierarchy, how it works. Like in an organization, we have different levels. Like we have the lower levels and then the people at the higher level and then the people at the management level. In the same way, the nervous system is very coordinated activity of the body. Now, how it works, this flowchart is very important. First of all, there's a stimulus. What is a stimulus? Stimulus is any change in the external or the internal condition that makes your body to take any action or produce a response. For example, suddenly the temperature increases. So what you'll do, you will wear lighter clothes, you will switch on the fan. So you take some action because you want your body temperature to be normal. So first of all, there's always a change. So change could be change in temperature, pH, salt and water level. Now this change is detected by the body. Now who detects this change? These are the receptors. Now the change could be external and the internal. So the external receptors are your sense organs. So for example, there's a change in light intensity, the eye will detect. Change in sound intensity, your ears will detect. Change in temperature, your skin will detect. Or there are internal receptors in the brain, because at times what happens, the outer temperature is normal, but suddenly your body temperature increases. So there are changes in the internal conditions which are detected by the receptors in the brain. Now, when they detect the change, they send the message to the controlling center. The controlling center is the central nervous system, which composed of brain and the spinal cord, which takes the information from the receptors, process the information, decides the action, and sends the message to the effectors. What are the effectors? Effectors are the muscles of the gland, because your body, to control, to make the stimulus, to respond to the stimulus, they need to either take any action, that is to move any muscle, or they need to release a hormone. So the effectors are generally the muscles of the gland, which are directed by the central nervous system to take actions, and then they produce a response. For example, there's a change in blood glucose level. So your brain receptors will detect it, goes to the central nervous system. Central nervous system directs the pancreas to release insulin. Okay, so effectors in this case would be the gland. Now let us look at what are the three types of neurons that we find in the nervous system. The first type is the motor neuron, which takes the impulses away from the CNS, which was here. So motor neurons were sending the message from the central nervous system to the effectors. And if you see this, it has a long dendrite. So if you remember in the cell biology, we did that. It's a special cell. It has a long dendrite that receives impulses and they transmit it through the long axon. And then at the end, there's a nerve ending. Then there's a sensory neuron, which takes the impulses to the CNS. It takes the message from the receptors to the central nervous system. It has a small cell body and the long axon. And then the dendrite is also extended. And at the end, you have the nerve ending. Then you have the relay neuron, which are found in the CNS. That is, these are the neurons which are found inside the brain and the spinal cord. So they connect the sensory and a motor neuron. So they just have a big cell body, does not have a long axon, as the impulses do not have to travel a long distance, and they have the nerve ending. So you should know the difference between the three types of neurons, where we find them and what is their function. And if possible, you should know this diagram of the neuron and what are the labelings, what are the each terms, each things which are there in this structure. Next is a reflex action. At times you must have seen that you touch something hot, you immediately remove your hand, and after you have removed your hand, you realize that you have taken any action. So you take that action which is automated without even you thinking, because your body receptors has already detected it and found that it's an emergency situation. And what do? They send the message to the spinal cord and not to the brain. So there's a stimulus detected by the receptors. The receptor sends the message via sensory neuron to the spinal cord, remember, not to the brain, and then the effectors and produces a response. So you can see here it's a spinal cord. It's like a head-shaped structure. And if you touch something sharp or hot, the message goes by receptors, via sensory neuron to the spinal cord. Spinal cord sends the message to the relay neuron. The relay neuron sends it to the motor neuron and to the muscle to remove the hand. So it is the automatic response of the body to a stimulus. In reflex action, the message from the sensory neuron is passed to the spinal cord instead of brain. The spinal cord sends the message to the effectors and produces a response. Example, knee-jerk reflexes, touching hot objects, sudden closure of the light with bright light. The advantages of the reflex action is it is rapid, it is quick, it is automated, instantaneous, without any conscious thought. Okay, so I hope this concept of reflex action, how it works, and the advantages are clear to you. Next we have is a synapse. Now, you know that the message in the neurons is transmitted by electrical impulses. But there comes a point, which is this, which is a junction between the two neurons. And this junction between the two neurons, or the gaps between the two neurons, is the synapse. And here the message is not transmitted electrically. It is transmitted by the chemicals called the neurotransmitters. So what we have is, at the end of one of the neuron, we have the vesicles, which contains this neurotransmitter. Whenever the electrical impulses arrives, these vesicles are forced to fuse with the membrane, release the neurotransmitters into the synapse. This neurotransmitters is taken up by the receptors of the other neuron, and it produces electrical impulses on the other neuron. Okay, so you should know what is a synapse and how synapse works, and this diagram is important. Okay, so next we have the biggest controlling center of the body, which is the brain. So in the brain, you should know this is a hypothalamus. Then the main part is the forebrain, and then here you have the midbrain, and then you have the hindbrain, which is your cerebellum. The forebrain is cerebrum, and then you have the stalk, which is your medulla oblongata. Now the cerebrum, which is your forebrain, is responsible for your consciousness, memory, intelligence, and the language. The cerebellum, which is the hindbrain here, which is a round circle, it is responsible for muscle coordination and balance. And this long stalk, which is the medulla oblongata, is responsible for unconscious activities like the heart rate, the breathing rate, and the gut movement. So you should know the labeling of each of these parts along with the function. Now, the brain scan is where we do the magnetic resonance imaging and take the images of the different parts of the brain, and then we find if there's any clot, if there's any thrombosis, if there's any problem there with the brain, and then we relate this problem with the loss of function of the individual. Now, there are a lot of problems with the brain scan because the brain is complex. There's a skull which protects the brain. There are thousands of neurons, thousands of neurotransmitters involved. The function of the different parts is still not known, and due to the presence of skull, it is difficult for the drugs to reach the brain. So the brain damage is the biggest problem. Okay, therefore people are advised to cover and protect the heads while they are driving to make sure the damage to the brain is protected. Okay, next is the eye. This is the structure of the eye. You should know different labelings and the function of each of them, and it is very important. So the front part you can see here is the cornea. It's like a little bulge outwards, and the function of the cornea is to refract the light so that it can enter the lens properly. Then in front of the eye, you can see this is an iris. This is like a muscle that controls this aperture, which is the pupil. Okay, a pupil is nothing but it is the aperture which opens and closes depending on the light it enters. If the light is not that bright, the iris will retract back, increasing the hole so that more light enters. If there's a bright light, then the iris will decrease the size of the pupil. Then you have the lens. The lens function is to focus the light on the retina. Then the lens is attached with the help of the ciliary muscles. It can contract and relax to change the shape of the lens. And there's a suspensory ligaments that's a support the lens onto the ciliary muscles. Then you can see there is a retina. Retina is the place where the image is formed, and then you have the choroid layer, which is the blood vessels that gives all the nutrients required to the eye, and there's a fovea, which is the part of the retina only with the cone cell, and then there is a blind spot. This blind spot is where the optic nerves enters the retina, and this optic nerve sends the message from the retina to the brain so that it can process the information and helps you to see. And then you have the fluid that fills the anterior part, which is the aqueous humor, and the posterior part is filled with the vitreous humor. This is the fluid that fills the eye and helps the eye to withstand pressure. Okay, so I hope all these parts along with this function is clear to you. Now, eyes do different changes so that it can see the far objects and the near objects, and these changes are known as accommodation. So for a distant vision, you can see that the ciliary muscles relax, making the suspensory ligaments tense, which in turn makes the lens thin. So you can see to see the far points, the lens has become thin so that the image is properly focused onto the retina, and the retina then can sense the message to a brain via optic nerve. But when you see the near point, the ciliary muscles, they are contracting, making the suspensory ligament too slack so that the lens become thicker so that the image can properly be focused on the retina. So you should know what changes takes place in the lens for distant vision and the near vision. Now, there are a lot of defects of vision. At times, people can just see the objects which are just at a short distance and cannot see the objects which are at the long distance. So there's a short-sightedness. So there's a short-sightedness and the long-sightedness. So in short-sightedness, which is myopia, the image falls, you can see, in front of the retina so that you cannot see it properly because the eyeball gets elongated. You can see that. So for that, what we do, we use a concave lens which properly refracts the light and make sure it falls on the retina. In hyperopia, you need to remember the image falls behind the retina. So the eyeball gets shortened and it is corrected by a convex lens. So you should know these both defects of vision. Now, there are other new eye technologies that have emerged. Now we can take contact lenses because it's not always feasible for the sports player or for the models to wear the spectacles all the time, the concave lenses or convex lenses. So in that case, we put the lenses just on the surface of the eye, which can be soft, self-disposable, and they can be used by any person at any age. Then there's a laser surgery in which we change the thickness or the curve of the cornea so that it can properly refract and make the image at the retina, but it is generally done at the adults because at the growing stage the people can still have problems, so laser surgery cannot be successful. Then there's a replacement lenses where you either replace the faulty lens or insert the correct one in place of the faulty one. Now there are some damage risks because it's a surgery, so there are some damage risks to the eye that is involved. Okay, so I hope these all defects of vision and what are the treatments is crystal clear to you. Now we have seen the hormonal control. Now let us see the nervous control. So to remind you, we started with this concept of homeostasis, and we saw that homeostasis involves the nervous system and the endocrine system. The nervous system uses the system of the neurons, and the endocrine system involves the endocrine glands, which releases the hormones to bring the body back into its normal levels. Now, what is the difference between the hormonal and the nervous system is that the hormonal system releases hormones, which are chemical messengers secreted by the endocrine glands. They are secreted into the blood, travel to the target organ. The target organ has receptors, and hormones bind to the receptors and triggers a response, and it produces a slower but a long-term response. On the other hand, the nervous system uses a system of neurons which sends electrical impulses to produce a response. The message is transmitted via electrical impulses. The response produces localized, and impulses do not travel large distances, but the response is quicker but short-term. So you should know this difference between hormones and nervous, how it works, and which one is more quicker and effective. Now there's a master gland. The master gland is a pituitary. Why it is a master gland? Because it controls the other gland of the body. It controls the follicle stimulating hormone, which are responsible for management of menstruation and reproduction. Antidiuretic hormone is also controlled by the pituitary gland, which maintains the water level of the body. And thyroid stimulating hormone is also controlled by the pituitary, which maintains the metabolic levels of the body. Okay, so it's very important to know that pituitary is a master gland, and why is it a master gland? Now, to explain why is it a master gland, you should know what hormones it control and how it works. So pituitary controls the follicle stimulating hormone, which works at the ovaries and makes the female sex hormones estrogen, and that is responsible for the secondary sexual characters and the development of egg and the menstrual cycle. Thyroid stimulating hormone released by the pituitary works on the thyroid gland, stimulate the glands to release thyroxine, and the thyroxine affects liver and kidneys and controls the metabolism. And the antidiuretic hormones targets the kidney, which controls the water level by causing reabsorption of water. Adrenal gland releases adrenaline, which targets liver and heart and prepares the body for fight and flight. Testes releases the hormone testosterone, which targets the male reproductive organ and develops secondary sexual characters. Pancreas releases insulin and glucagon. The target organ is the liver, and the insulin decreases the blood glucose level, and the glucagon increases the blood glucose level. Ovaries releases estrogen and progesterone, which targets the female reproductive organs, controls the development of the egg, menstrual cycle, and develops secondary sexual characters. So these are some of the examples of the hormones with their glands, their target organs, and their effect, and you should remember them all. Okay, next we have is how the blood glucose level is maintained in the body, and this is an example of glucose homeostasis. Now, this works by the pancreas, and the pancreas releases two hormones called insulin and glucagon. Glucagon, you need to remember. Insulin lowers the blood glucose level, and glucagon increases the blood glucose level. So after meals, what happens? The blood glucose level suddenly increases. So what does pancreas do? It releases insulin. Now, insulin has these effects. It increases the permeability of the cells to take glucose so that more glucose enters the cell so that blood glucose level can go down. If the body does not require any more glucose, it converts all the excess glucose into a storage carbohydrate, which is a glycogen. It also converts the glucose into the fats and stops the breakdown of fat so that all these factors can result in decrease in glucose level.
So this is how the glucose metabolism works. When the blood glucose level rises, the pancreas releases insulin. The insulin does all these factors and makes the blood level, glucose level to be normal. On the other hand, if the blood glucose level is too low, then the pancreas releases glucagon, and the glucagon does all the things opposite to insulin. So you need to remember glucagon is the hormone which increases the blood glucose level, and glycogen is the stored carbohydrate which is made in the liver, where excess glucose is converted into glycogen. Now, what happens if your body glucose level is not controlled? When the body glucose level is not controlled, it can lead to diabetes, and there are two types of diabetes, type 1 and type 2. Type 1 is insulin dependent, as in this case, the body does not produce insulin. Type 2 is insulin independent, because in this case, body do produce insulin, but your body is not responding to it. Your body becomes resistant to insulin. So in type 1, it is caused by damage to pancreas, or by some autoimmune disorder, or by accident there's a damage to the pancreas. Type 2 is caused by poor lifestyle and diet. In type 1 diabetes, the person can be given insulin injections. In type 2, it is treated with lifestyle changes and a balanced diet. Type 1 is most common in young ages, where type 2 is common in obese people. Type 1 can be genetic, and type 2 is mostly environmental. In type 1, drugs might not be required. We just require insulin injections, whereas in type 2, there are some drugs are taken to make the body to respond to insulin. Okay, so you should know the differences between the type 1 and type 2. So what are the treatments for type 1 diabetes? Insulin injections. Remember, we need to take insulin injection. We cannot take insulin tablets, because insulin is a protein hormone, and protein can be digested in the stomach. The insulin goes to the blood, and it converts the excess glucose into the glycogen and control the blood glucose level. In type 1 diabetes, people are asked to take less carbohydrates, and if in case there are a lot of problems in managing that, pancreatic transplant, pancreatic stem cell transplant, or using stem cell to regenerate pancreatic cell are some of the future cures for maintaining type 1 diabetes. For type 2, balanced diet, regular exercise, weight management, taking drug to increase sensitivity of pancreas to insulin, or taking more insulin injections with the thought that increased concentration might increase responsiveness are the possible management techniques to maintain the type 2 diabetes. Okay, so you should know the difference between both the types of diabetes and how we control both of them. Now, this insulin maintenance, or the maintenance of water level, which we'll be seeing later, is an example of negative feedback. What is negative feedback? Negative feedback is when the level of anything rises above optimum, like the glucose concentration, the body tries to decrease it. And if the level of anything decreases below optimum, the body tries to increase it. So body is always doing negative or opposite to what has happened, and this is known as a negative feedback. For example, if the blood glucose level goes down, then the body releases glucagon that increases the blood glucose level. If the blood glucose level goes up, the body releases insulin to decreases the blood glucose level. Okay, so I hope this example of negative feedback and what is negative feedback is clear to you. Now you should know there's another hormone called adrenaline, which is a fight or flight hormone. It is a stress hormone, emergency hormone. What effect it does to your body? It increases your heart rate, it increases your breathing rate, it increases the blood flow, it dilates the pupil, it diverts the blood flow away from the gut, and it increases the blood flow to oxygen to the brain, so that you become more alert and take quicker action. Okay, so you should know the effects of adrenaline. Now, next we have to do the human reproductive system, the male reproductive and the female reproductive system. The male reproductive system, it consists of a penis, there's a urethra, there is a bladder, there's a seminal vesicle, there's a prostate gland, and then there's a testis which produces the sperm, which is there in a scrotum. The sperms are released to these sperm ducts, and the seminal vesicles, it releases semen which mixes with the sperm, and it goes out through this urethra. And this male reproductive organ, testes, it releases a hormone called testosterone, which is responsible for secondary sexual characters. And the female reproductive system is the system of the two ovaries, and then there's a fallopian tube. The ovaries releases the egg into the fallopian tube, which travels into the uterus, and then there's a cervix and the vagina. And the female hormone is the estrogen, which is released by the ovaries, and that is responsible for the secondary sexual characters. Now, it's a very important hormonal mechanism in the body, which is a menstrual cycle, which is purely hormonal. Now, menstrual cycle is a cycle for 28 days, and in these 28 days, these are the changes that takes place in the body. So you can see this in this diagram. From day 1 to 5 is where the bleeding stage happens, which is the menstrual stage. There's a shedding of the uterus lining, and you can see all the hormones, they are at the lower level. From day 5 to day 14 is the egg maturation phase. So that is a follicular phase where egg is matured in the ovary, and the hormones responsible for the maturation and development of the egg is the follicle stimulating hormone. So you can see that the follicle stimulating hormone is started to increase in concentration. Then at day 14, the egg is released into the fallopian tube, and this egg is released, and this release of egg is caused by a hormone called luteinizing hormone. So you can see there's a sudden surge of this luteinizing hormone at the day 14. Now, after day 14 to day 18, there's a luteal phase in which there's a sudden increase in the progesterone and the estrogen hormone, which maintain. So there's an increase in progesterone and the estrogen level, because progesterone and estrogen are responsible in maintaining the uterus lining. So they maintain the uterus lining, they make it thick so that they can support the fertilized egg and cause the pregnancy. But if in between 14 and the 28 days, the egg is not fertilized, then the level of the progesterone suddenly falls, and it leads us to the shedding of the uterus lining, causing menstruation, and it starts with the day 1 to day 4. Okay, so you should know what are the changes that takes place in all these days, what is the phase, and what are the hormones that are involved. So what are the hormones that are involved in menstruation? These are pure hormones: follicle stimulating hormone, luteinizing hormone, estrogen and progesterone. Follicle stimulating hormone and luteinizing hormone are released by the pituitary, and they are required in the first 14 days. Day 1 to day 14, it's the follicle stimulating hormones, which causes the maturation of the eggs in the follicle and stimulates the production of estrogen, and the luteinizing hormone causes ovulation. Then the estrogen is released, stimulated by the FSH, and from day 14 to day 28, it develops a uterus lining and stimulates more LH but inhibits FSH. And the progesterone is released by the empty egg follicle in the ovaries, and it is responsible for maintaining the uterus lining from day 14 to day 28 and prepare for pregnancy. It inhibits both LH and FSH, so no menstruation happens during pregnancy. Okay, so I hope these various hormones of menstruation are clear to you. Now, to prevent fertilization, there's a contraception method, and the contraception method, they either prevents the sperm to meet with the egg, or if the sperm meets the egg, they prevent the implantation of the fertilized egg into the uterus, thus preventing the pregnancy. Now, this can be achieved by the barrier method. The barrier method includes using diaphragms or condoms to prevent the sperm to meet the egg, or the hormonal method, which prevents the egg to mature or prevents the implantation of the eggs in the uterus, or the chemical method, which uses spermicide to kill the sperms, or the intrauterine device, which prevents the embryo from planting, or surgical method, which provides permanent contraception. Okay, so these are the various contraception methods, and they can come in a long note maker or a small question. So you should know what are the various contraception methods that we have. Next we have is how contraception works. So in contraception, there are 3 major techniques involved: the contraceptive pill, the intrauterine devices, and the surgical method. Now, what are contraceptive pills? The contraceptive pills contains mix of female hormones, estrogen and progesterone. We call them mixed pill. They prevent the release of FSH, so thereby they prevent the maturation of egg. They also make thick mucus in the cervix, which prevents the entry of the sperm. They also prevent the uterine lining development, so they prevent the implantation. Some pills, they just contain progesterone, and at times the contraceptive implant is also inserted. So there you don't have to take the pills regularly, and this implant slowly releases progesterone and maintains the uterus lining. Or the contraceptive patch is also used, which absorbs and mixes the hormones into the blood. They have these side effects like blood pressure, they have to be taken daily, and they cause changes in the menstrual pattern. On the other hand, intrauterine devices are the mechanical devices like copper T, which is inserted into the uterus. It releases copper ions, which are toxic to the sperms, and this device also prevents the implanting of the embryo into the uterus. And some of these IUDs, intrauterine device, they also releases progesterone, which works the same way as the progesterone pills, which prevents the release of FSH, preventing the maturation of egg. They make thick mucus in the cervix to prevent the entry of sperms, and they also prevent the uterine lining development, preventing implantation. But IUDs have side effects like infections or internal bleeding. Then we have the permanent contraception, which are mostly taken by the people who already have kids. They do surgical methods, which is vasectomy, which is the male sterilization, and tubectomy, which is the female sterilization. In vasectomy, the sperm ducts are cut and sealed so that sperms cannot enter the urethra, which prevents fertilization. And in tubectomy, the oviducts are cut and tied to prevent the release of egg, which prevents the fertilization. The side effects of this is it is permanent, so you cannot resume your fertility back. But with contraceptive pills, you can stop the pills and the hormone levels can be normal. And in the intrauterine devices, you can remove the device and then the fertility is restored. Now, there are a lot of infertility problems that happens in people. The first problem is the ovulation problem, where the eggs do not mature or the eggs is not ovulating. In those cases, the women are given fertility drugs, which are mix of FSH and LH. So follicle stimulating hormones causes the development of the follicles, and luteinizing hormone stimulates the ovulation. At times, there's a tubes that is faulty, so that the embryo is not able to come to the uterus, or there's an implantation problem. In that, we perform an in vitro fertilization where embryo is developed outside and it is implanted back into the uterus for the development of the pregnancy. And in the third case, even implantation happens, but pregnancy development doesn't take place. In that case, there are problems with the uterus. So the surrogate mother, where the fertilized egg is implanted into another mother who gives birth, can be a solution. Now, what is in vitro fertilization, which is very promising, and that has given babies to many of the people who do not have babies. So what do? Is we first give fertility drugs and stimulate ovulation. Then we collect the egg and the sperms, and we put it into a petri dish, and we cause the fertilization to occur. When the fertilization is occurred, we cause the zygote to divide and develop into the embryo. The embryo is then taken and inserted surgically into the uterus lining. Okay, and then this develops into a baby. So this is what is the concept of in vitro fertilization, but it has problem as it is expensive. It can result into multiple embryos. It can result into premature birth. It can result into birth with disability, and it cannot be always successful. But that has caused, it has shown a good success rate, and there are a lot of people who are opting for in vitro fertilization. Next is plant hormones. As in humans, there's a hormones controlling the body mechanism. In the same way, plant also needs some survival changes. For example, they should grow towards light. The root should go towards the gravity. So these are controlled by the plant hormone. So there are two important movements that we see in the plant is phototropism, where the shoot moves towards light. You can see in this example, the shoot is moving towards light, and there's a gravitotropism where the root moves towards the gravity. Now, why the shoot moves towards light? It is caused by auxin. When auxin is produced in the root tip and the shoot tip. So when the light falls on the auxin, you can see auxin has shifted towards the shader side. And when the auxin shift towards shader side, auxin causes the growth or the cell elongation of the shader side. As a result, cell divides onto the shader side, causing the shoot towards bend towards light. On the other hand, in response to gravity, the oxygen shift towards the grounder side. So towards the lower side, the oxygen develops in response to gravity. And in the roots, oxygen has an opposite effect. Where it is concentrating, it is inhibiting the growth. So the other side of the root grows, which results in the bending of the root downwards. So oxygen causing bending of the root downwards and it causing the shoot to grow upwards. Why? Because in phototropism, oxygen is causing the growth in the shader region, and in the gravitotropism, the oxygen is inhibiting the growth onto the side where it is present. So you should know how both of these mechanisms works. Now there are 5 important hormones in the plant which are very important: oxygen, gibberellin, ethene, cytokinin, and abscisic acid. Oxygen stimulates cell division and is responsible for growth of the plant. It is used in the tissue culture as a rooting powder as it causes the root to develop quickly. And it is also used as a weedicide because when the weed, due to its broader leaf, take the oxygen quickly as compared to our main crops, and these concept of thermoregulation as what changes takes place in the body when suddenly the temperature becomes too low. So when the temperature becomes too low, you must have seen the body starts to shiver. Why? Because the body detects that there is a sudden decrease in the body temperature, so the heat needs to be generated. So shivering increases the heat so that the body temperature can raise to normal. Second, the thyroid stimulates cells to increase the metabolism so that the heat production can be increased. You must have seen you get goosebumps in cold. Why? Because the body hair stands up so that they can trap the layer of insulated air which can prevent heat loss by conduction. And you must have seen your body tends to get blue. Why? Because of the vasoconstriction. So when the temperature becomes too low, the capillaries that is supplying the blood to the surface of the skin, they constrict. They reduces the blood flow to the surface. As the blood flow to the surface is reduced, the blood can lose less heat due to radiation. On the other hand, when it gets too hot, the body hairs lies flat. There's a sweating. The sweat glands produces sweat. The sweat causes cooling of the body by evaporation. The metabolism of the body decreases. So the thyroid stimulates cells to decrease metabolism so that the less heat can be produced. And then there's a vasodilation. What is vasodilation? The capillaries that are providing the blood vessels of the capillaries that supplies the blood to the surface dilate so that more blood flows to the surface. And when more blood flows to the surface, it will lose heat by radiation. So in vasodilation, or in the hotter times, we are making more blood flows to the surface. So you should know what is the difference between vasoconstriction and vasodilation and what changes takes place in the body in response to temperature. Okay, now let us see what are the different waste products produced in the body. Waste products are the products which are produced by the metabolic reactions like respiration and digestion. The first waste product is carbon dioxide produced during respiration. It is toxic because carbon dioxide can disturb the pH and affects the enzyme activity. So it needs to be removed immediately. So it is excreted out through the lungs. Then there are water. Then by metabolism, we also produce water like in respiration or during the digestion process. It is excreted through the skin in the form of sweating or breathing, or kidneys in the form of urine. Water can also disturb the osmotic balance and salt levels. So it is very important the level of water is also maintained in the body. Now when there's excess carbohydrates, excess fats due to digestion, that can be easily stored in the body. But protein is very harmful. Excess protein cannot be stored in the body. So the protein is digested into amino acid and the liver deaminates. That is, by the process of deamination, converts amino acid into ammonia, and ammonia is converted into urea. So urea is another waste product produced in the body by the metabolism of protein, and this urea is excreted by the kidneys in the form of urine. Okay, so how these kidneys function? So we have two kidneys, and these kidney receives the blood, they filters the blood, and they pass the urine into via these two tubes called ureters into the bladder. The bladder stores the urine and expels the urine out through urethra. Now there are two systems that works in the excretory system, the ultrafiltration and the selective reabsorption. In ultrafiltration, the kidneys filters the blood at a very high pressure. All the waters, glucose, and useful components get also into the kidney filtrate. The blood cells and the blood proteins are too big size to be filtered. Now, once these essential things are there into the kidney filtrate, they need to be reabsorbed back into the blood. So this process is known as selective reabsorption, where the useful components are reabsorbed back into the blood. The waters get reabsorbed at different levels depending on need of the body. Therefore, we call it a selective reabsorption. Now let us see how the body regulates the water level. And the regulation of water level is known as osmoregulation, and it is also a negative feedback. And it involves the release of a very important hormone called an antidiuretic hormone by the pituitary. So when the water level decreases in the body, it is detected by the osmoregulatory centers in the brain, which directs the pituitary to release a very important hormone called the antidiuretic hormone, which travels into the blood and enters the kidney tubules, and it increases the reabsorption of the water into the blood by increasing the permeability of the kidney tubules. As a result, more concentrated urine is passed, and it also promotes thirst, so that you take in more water to resume the blood water level. On the other hand, when the water level increases, it is also detected by the osmoregulatory centers in the brain. The release of ADH in this case is inhibited by the negative feedback. So there's less reabsorption of water, so more dilute urine is passed. So you should know how this mechanism of osmoregulation works. Now there are people whose kidney get damaged either by an autoimmune or a genetic disorder or by some accident. So in this case, they have two solutions. First, either they take the help of the artificial kidney called the dialysis, or they do a kidney transplant. Now what happens in a dialysis is the patient blood is taken out and it is given a heparin, which prevents the blood clotting, and then the blood is fed into a dialysis fluid, a dialysis machine which has a dialysis fluid. The dialysis fluid contains no concentration of urea and the ideal concentration of minerals and the salt level as that of the blood, and the dialysis tubes are made up of the semi-permeable membrane. So as the blood flows into the dialysis fluid, the urea is diffused out along the concentration gradient, and excess salt is also removed, maintaining the normal salt and mineral level and absorbing all the urea into the dialysis fluid. The blood now becomes pure and clean and is pumped back into the patient. Okay, so in the artificial kidney, the blood flows into the dialysis machine, which contains the dialysis fluid. It contains the same concentration of essential mineral ions as that of the blood, but no urea. So as the blood flows into the dialysis fluid, the urea is diffused out along the concentration gradient, and excess salt is also removed, maintaining the normal salt and the mineral ion level, and the clean blood is then pumped back. The problem with the dialysis is that you need to regularly visit the hospital, you need to have the lifestyle changes, the changes diet and regular expenditure are some of the disadvantages. On the other hand, there's a kidney transplant. You can see there's a damaged kidney. So from a donor, a normal kidney is transplanted, and that works normally as like of the kidney. So the donor has to be a close match, because the body can reject the donor kidney, because it can take that as a foreign antigen and produce antibodies and destroy it. So what do we do in this case is the person has to be given an immunosuppressant drug so that the body immune system does not reject it. But the problem is it does not last longer, and since the person is on immunosuppressant, the person can get prone to infectious diseases. Okay, so there is a trade-off. So which is the best way, dialysis versus kidney transplant? It depends on the patient condition, what he is willing and what he's not willing to do. Because dialysis has an advantages of getting no surgery, no infection, no immunosuppressant drug, and easily available. But the disadvantages of lifestyle changes, regular visit, restricted diet, and a long procedure. On the other hand, kidney transplant does not require any lifestyle changes, no diet restriction, no regular visit, but it does not last longer. There are chances for rejection. There are immunosuppressant drugs need to be taken. The person is more prone to infection, and finding a suitable donor is a big problem. So the promising future for the kidney disorders is using stem cells, the person's own stem cells, to create the kidney cells or the nephrons, so that the problem of rejection can be avoided and it can be a long-term solution. But again, the development and the research is still going on. Okay, so I hope this topic, a long topic with a lot of key terms, is crystal clear to you. Now let us see what are the key terms you should know. So you need to write about all these key terms, pause the video, write about these key terms, and come back. So you should know what are homeostasis, receptors, effectors, stimulus, neurons, central nervous system, sensory neuron, motor neuron, relay neuron, reflex arc, brain, cerebral cortex, cerebellum, medulla, MRI, eye, cornea, retina, blind spot, ciliary muscle, suspensory ligaments, sclera, iris, pupil, accommodation, myopia, hyperopia, endocrine system, hormone, adrenaline, insulin, pituitary gland, FSH, LH, estrogen, progesterone, glucagon, glycogen, diabetes, menstruation, ovulation, IVF, phototropism, gravitotropism, oxygen, jubilin, ethene, vasoconstriction, vasodilation, thermoregulation, dialysis, and selective reabsorption. Okay, so I hope you're able to answer these key terms. If you still are not able to answer, go back to the video, look over these terms again, and you can visit the website and check the explanation of all these. Okay, so I hope this topic is clear to you. As always, the next step, check the specification, make sure everything which is there in the specification is crystal clear to you now, and you should do exam question on this topic, which can be easily found on my website. The link is shared with you in the description box below. All these notes are there on my website. You can click on them and read them for quick revision. If you like this video, please do subscribe to my channel, and do not forget to like, comment, and share this video. And also click on the bell icon, because if you click on the bell icon, you'll be notified as soon as I put a new video. And if there's any specific video you want me to put a lesson on, please leave a comment below, and I'll make sure I'll have that up before your exam. In case you have any doubts in any of these concepts mentioned in this video, leave a comment below, or you can come to my website where we have a 24/7 chat support till your exam to answer all your queries. Okay, so I'll see you next in my next video. Till then, happy revising.